Good morning, good afternoon, or good evening, everyone. First, I would like to thank Dr. Ye Mengchen and the organizing committee for their very kind invitation to allow me to share some of my thoughts at this very important conference. Traditional Chinese medicine, TCM, has been my passion for 4 decades. I feel the urgency to modernize it at this critical time when biomedical research blossoms with big data. The argument that have been around for decades is if the methods of modern science can be applied to TCM without losing TCM's identity. Here, I try to put forward some of my thoughts for a TCM expert to consider. 20 years ago, individualized treatment was the hallmark of TCM. However, today, 2021, individualized precision treatment has been used in modern medicine. One good example was the US FDA announcement on August 15, 2019 for the third cancer drug that targets a key genetic driver of cancer. In addition, you can also find 3 tables of pharmacogenetic association information with 111 listings on US FDA's webpage. Stepping into the 21st century, advance in biotechnology is generating huge amounts of data, such as in the area of genomics, transcriptomics, metabolomics, and proteomics. Individual human differences have been studied at level of single nucleotide variation and single gene mutation. Many new studies focus on disease heterogeneity in human population, such as type 1 diabetes or hypertension or heart disease. Samples collected long time ago in large cohort that have continued for many generations are reanalyzed with omics assays. All these efforts generate huge amounts of data and create many valuable data resources. In addition, advance in single-cell technology has enabled scientists to zoom in to rare population of cell types that contribute to disease and to zoom out to understand the full landscape of cell types in organ and tissues, which have never been done before. All these big data studies not only greatly enhanced our understanding of biology, but also stimulate the development of new data analysis methods. These new methodologies are key to address human heterogeneity at molecular level. Let's look at the examples of exercise studies to understand the implication of human heterogeneity and the challenge of N of 1. Exercise is a behavioral intervention. It's different from the effects of single compound drugs that have a clear biological targets and with a less variation among human population. Exercise stimulates body's systemic adaptations that have very high variability among human population. This type of natural behavioral stimulation has similarity with acupuncture treatment, which also has wide range of responses. These figures summarize the distribution of exercise response. As you can see, the exercise response in human population were not uniform at all. The non-uniformity of exercise response in humans was also shown in this multi-omics study published last summer by Dr. Mike Snyder. In this study, thousands of genes, proteins, and metabolites were changed after only 10 minutes of exercise. A very interesting finding of this study was highlighted in the figure D that really related to TCM N of 1. This is a t-SNE plot, which generated by a nonlinear dimensionality reduction algorithm used for exploring high-dimensional data. It maps multiple dimensional data to 2 or more dimensions suitable for human observation. Each dot in this graph represents a single sample. Each color represents a single participant in a study. Samples were taken before and after exercise with multiple time points. As you can see, all the same color dots were closer to each other in this 2D t-SNE plot, which indicate that molecular feature of before-after exercise of individual participants were closer to themselves instead of before and after grouping. This was not the case for most of the animal studies, especially for exercise. Here is an example of a rat lifelong exercise study. Urinary metabolites were sampled repeatedly from the same rat. What was shown here is a discriminant analysis result. The dot represents each sample, color represents grouping. As you can see, exercise and non-exercise control were separate completely. This is because this particular study used inbred rat, and they are genetically identical. Because human is different from inbred animal, data analysis need to be done differently too. For human study, data from each individual need to be compared with his or her own baseline. The molecular features can be combined after extraction of differential features for further analysis, such as temporal dynamics, pathways, and networks analysis. In summary, the individual feature extraction step is critical for human study. Now, let's look at how TCM manages human heterogeneity. TCM starts with collection of list of TCM symptoms from a patient. Then TCM doctor, based on TCM theory, makes individualized syndrome differentiation. Based on TCM syndrome, treatment plan were made. More importantly, for TCM, the process does not stop here. The outcome of the first treatment plan is circled back as the beginning of next cycle. From the point of view of dealing with heterogeneity, both omics approach and TCM are very similar. They both have the critical step of clustering N of 1 cases. The critical difference are one based on computational statistical algorithm and the other is based on expert experience and TCM theory. In reality, TCM is much more complicated. One of the critical features of TCM is expert dependent because there were so many critical decisions need to be made by the masters. Although the actual phenotype accessed by TCM is objective, however, the assessment itself is very subjective. In addition, there were so many layers and directions of many-to-many relationship. The complexity of TCM is like a complicated hairball. Over the years, TCM have developed its own algorithm to deal with complexity. Here are 3 examples. When dealing with infectious disease, TCM use human response to weather changes instead of using invisible organism types. When dealing with drug herbs, TCM using natural taste that human has instead of using chemical structures. When naming acupuncture point, they're often used, in many cases, they use human response to acupuncture instead of anatomical locations. All these are really pointed out that the focus of TCM algorithm is on human body, on human response to whatever perturbation, internal or external. This smart strategy is also a natural strategy evolved with the survival of human as a species from natural infection. Here an example of an innate immune system that using similar strategy. TLR is called toll-like receptor. It was first identified in fruit fly and was found to be an important immune sensor that was evolutionarily conserved. This TLR system is the gatekeeper of immune defense system. There are millions of pathogens or even immune, internal wastes that can be detected by this system. There are 11 different types of TLR receptors, and it able to detect 8 different classes of chemical structures and trigger 5 classes of immune responses. As you can see, from million pathogens to 5 types of immune responses, this is a very effective natural dimensional reduction system. There are many publications on herbal formula that actually tackle the immune responses specifically. There were studies on different types of diseases like infectious disease, asthma, autoimmune disease. As you can see, if we are able to manage our immune system by modulating that 5 types, we don't have to worry about millions of pathogens out there. So that's why I was not surprised when I hear that TCM herbal treatment have been contributed greatly in fighting SARS-CoV-2 infection. To leverage TCM success on managing human responses, I think there are 2 things are critical that we need to pay attention to. One is transform subjective to objective, from diagnosis to treatment. And the other is to increase resolution and the precision of TCM phenotyping capability. Exploring molecular and physiological biomarkers of TCM diagnosis and therapeutic effects can greatly enhance the precision of TCM therapies. This type of TCM biomarker discovery research can be done with N of 1 case of human patient study in TCM research clinic that collects before and after samples such as blood, urine, gut microbiome, in addition to TCM phenotypes. The collection of N01 cases, at least in thousands, can be used for multi-omics analysis. It is important that we also integrate the TCM doctor and herbal medicine as subject in this framework with the patient. Once unbiased high-resolution data are available, artificial intelligence technology can advance TCM to a new level. Without higher resolution data, feeding AI on current level of TCM data can only generate algorithms that replaces the role of TCM master at the most. Besides what I proposed above, people may consider a simple version of TCM biomarker discovery research, which is illustrated on this slide. In this case, patient will be diagnosed by TCM expert, and omics assays will be done on patient sample. Subsequent data analysis will be conducted to associate the omics signature with TCM diagnosis. What do you think about this design? Well, based on Dr. Snyder's human exercise study, this would be a very challenging task to get reproducible signals. Human heterogeneity will require to conduct such study in a very large cohort. Example of successful genetic association study were all enrolled more than 10,000 participants. I would like to suggest that in this type of study, having a perturbation and comparing before and after are essential elements to be considered. As I indicated in the beginning, I feel the urgency for moving TCM research to higher resolution. I show the achievement of precision medicine in 20 years. Another example as the precision medicine was a fast-growing research area called microbiome. NIH launched Human Microbiome Project in 2007. With less than 10 years, our knowledge on relationship between human body and the microbiome is completely renewed. Human body has at least 1-to-1 ratio of human cells versus microorganisms. Microorganisms as a whole serve as another important organs of human body. Now we know that microorganisms help us digest food, produce vitamins, and their metabolites regulates our body metabolism and control our mood. Microorganisms are also partner in crime for human diseases. This was a publication published in 2020 that summarized the herbal effects on gut microbiome. This is a clinical trial published in April 2021, showed clinical application of gut microbiome on treating child malnutrition. If the mechanism of herbal treatment is through modulating gut microbiome, there may be other methods that can be executed much more effectively than taking herbal medicine. TCM does not have another 10 years to prove it or optimize it. Now, I hope that you feel the same urgency as I do, and I hope that I have convinced you on following points. Modern biomedical research has been greatly accelerated with big data. Modern medicine is quickly filling the gaps where TCM has advantages. The big data technology is mature enough. It can be applied to explore TCM and help TCM to shine in the 21st century. With that, I would like to thank you for your attention. Here is my email on the slides. If you have any question, please don't hesitate to email me. Thank you.